my name is Joanne Ahern. I represent the BioDT project and I am the communication partner. We're delighted to bring this webinar to everyone today. We have Hanna Koivula, who will be giving the introduction to the data citation webinar, and then Daniel Noesgaard, who will introduce data citation the GBIF way. We will then have a Q&A and a conclusion at the end. The question and answers chat is open and you're all highly encouraged to use it. And then we also have the chat open for you to use. And I would like to welcome everyone who's today, who is from, we have many people from research institutions and universities. We also have many biodiversity research infrastructure nodes and data providers. These are the main people who have registered for the webinar today. We are delighted to have 166 people registered, which is wonderful to see. And they come from 60 countries worldwide. So it is really brilliant to see such a wide variety of people representing a wide variety of biodiversity environments as well. And I would encourage you, if you're interested in this topic, to watch the recordings from our previous webinars and follow BioDT to learn more about our journey. We have put the links on the slide here, but we will also copy those into the chat. And of course, myself and Gabriella, my colleague, are here to answer any questions you might have. Please just write to the two of us and we'll help you. And then I would also like to draw your attention to the second webinar in this series on data citation, which will take place on the 20th of November. And you can also register for that for free now. So that's just a very brief introduction from me, and you're all very, very welcome. Without further ado, I'm going to hand over to Hanna to introduce you to the BioDT project.
Okay, so fine. So that means
Hanna Koivula.
Thank you,
Hanna.
Joanne.
Thank you.
And I will share my screen. Now, hopefully you see it now in the presentation mode.
Yes, it is in presentation mode. Thank you, Hanna.
Excellent. So I was tasked to present what BioDT project is, and this presentation was originally prepared for TADWIC, Biodiversity Information Standard Conference, together with quite many co-authors. So I'd like to thank them for also helping with these slides. This biodiversity digital twin is a project building a prototype that will provide advanced models for simulation and prediction capabilities through practical use cases addressing critical issues related to global biodiversity dynamics. We have started June 22, and this is a project for 36 months, bringing together 22 partners that represent that are experts in biodiversity research, high-performance computing, artificial intelligence, digital twinning, and also FAIR principles. And you can find more information from biodt.eu. But what is a digital twin? Digital twin is a virtual representation of real-world entities, processes synchronized at a specified frequency and fidelity. Here, fidelity refers to the level of precision captured by the digital twin in comparison with its physical counterpart. So we have real space, which is the physical twin, and then we collect data of a phenomenon to create a virtual space, which is then the digital twin. And with the digital twin, it is possible to model and predict things which generate more information about the physical phenomenon. And then we can test whether this is true by then collecting more data and understanding more about the physical twin. And this reflection is the digital twin. So digital twin is composed of data and then a model that is the representation in terms of behavior and then an application that connects the data and model in a way that makes the outputs of the model relevant given the specific purpose of the digital twin. Since different scopes require different behavior and fidelity, there cannot be a single twin answering all possible questions. The BioDT project responds to key EU and international policy initiatives, including EU Biodiversity Strategy 2030, EU Green Deal, UN Sustainable Development Goals, and Destination Earth, which is a European Union digital replica of our planet project, which holds multiple different digital twins. The biodiversity digital twin brings data and services from four different research infrastructures, which are GBIF, LifeWatch, ELTER, and DISCO, and the research communities behind all these four at this point. The challenges and paths towards solution that we are aiming to answer with these use cases that are categorized into four different groups are metadata, which is needed for enhancing the data findability and giving it context. And then also important are the FAIR principles. Then we use controlled vocabularies for consistency and clarity in data interpretation. And then with these vocabularies, we can use also semantic mapping, which enables harmonization and meaningful integration of diverse data. We aim to utilize existing standards like Darwin Core and EML, which then serve as foundational networks. But combining biodiversity data, which includes events and evidence information with direct environmental measurements, present a multifaceted challenge. This integration is essential to understand how environmental factors influence biodiversity and the behavior of the models. As an example, we have like the forest digital twin is asking questions like, how will forest biodiversity change under different forestry and climate change scenarios? And how can these predictions be utilized in conservation and adaptive forest management? And by posing these questions to the digital twin application, we can try finding the most appropriate forest management strategies. And there will be a series of papers about these use cases and what have been learned from these modelings. So here as an example, one of them. I mentioned the semantic mapping, and this is something that links closely to also data citation. So foundation of digital twin application is data and models, but somehow we need to make those data and models and all workflows available to users to be transparent and reproducible. So harmonized abstraction layer using persistent identifiers, PIDs, and fair digital objects is being experimented. This is not very established techniques yet, so this is a pilot in itself. Semantic mapping and crosswalk techniques are needed to provide machine actionable metadata. Models in BioDT are formal representation of problems or processes implemented through equations, algorithms, or a combination of both. And a machine actionable metadata should answer following questions as an example. So what is the format of the input data? What types of data does the model accept? What is the format of output data? Where and how can the model be accessed? What types are needed to run and test the model? And what were the parameters used to generate a particular version of the model? And more information about these models and how we are constructing them, you can find from BioDT GitHub, and the link will be shared with the slides with the audience. So a digital twin requires that, or these data and models require robust computing infrastructure. BioDT is utilizing Lumi supercomputer for the prototype digital twins. The use cases also require a comprehensive suite of software tools and also need software for data acquisition, storage, processing, and analysis. Coordinating diverse software component is crucial aspect of BioDT. Also, workflows needs standardized procedures and protocols to be transparent and reproducible. And for this, we are implementing the so-called research object crate, RO crate, to organize and manage digital objects within different workflows. And this is something that has been already implemented with the Elixir infrastructure for the sequence data. Governance and stakeholder management is also very important here, and as the research infrastructures vary in governance approaches from top-down to bottom-up models, BioDT places a strong emphasis on aligning technical and data standards discussions among research infrastructures and stakeholders. This alignment fosters synergy, enabling the project to leverage diverse expertise and resources effectively. So as a conclusion, I could say that this is a groundbreaking and transformative approach to biodiversity research. BioDT leverages collaborative research infrastructures and partnerships like GBIF, DISCO, ELTER, and LifeWatch at this point. In the future, hopefully even more or wider variety of research infrastructures. And the foundation of BioDT rests on robust data standards and the ability to refer to multiple data originators and data components by using persistent identifiers. And with this conclusion, I would like to hand over to Daniel to talk more about how to use the PIDs and citate data and its components. Thank you.
Thank you, Hanna. Let me see if I can start my screen sharing. Okay, I hope you all can see that.
Yes, it's perfect.
Excellent. Okay, so yes, thanks to the BioDT project for organizing this webinar on data citation. I think it's a very important topic and one that I've been involved in for quite some time. So my name is Daniel Noesgaard, and I work for the GBIF Secretariat. My main responsibilities include ensuring that GBIF-mediated data is tracked and documented in order to be able to assess the impact of the data. And important to exactly that process are data citations, which is what I'll be talking about today. What they are, what their purpose is, and how to do them properly. And since I work for GBIF, I will be using that as the main use case for my talk. So let's start by looking at why we cite in the first place. So we use citations in publications when we rely on other people's work. And when I say work, I don't mean just journal publications. Datasets, and in particular, biodiversity datasets, take a lot of work to compile, and that effort needs to be acknowledged. So when you use biodiversity datasets in your research, you should cite them in the same manner as you would cite a paper. And such citations should always include unique persistent identifiers, such as DOIs, whenever possible. If we take a step back to where it all began, if you will, I've chosen to highlight a few significant events. In 2009, the organization DataCite was founded. DataCite is a DOI foundation registration agency with the aim of providing DOIs to datasets. And this is opposed to Crossref, who is also a registration agency, but Crossref focuses on journal articles. And GBIF is a member of Datasite and used Datasite for issuing DOIs. In 2014, a working group under Force 11 developed a joint declaration on the principles of data citation, which in effect outlined how data is legitimate, citable products of research, and that data citations should be accorded the same importance in the scholarly record as citations of other research objects, such as publications. And these principles have since been endorsed by 125 institutions, including publishers and funders. GBIF started assigning DOIs to all datasets in 2015, allowing them to be cited according to these principles, basically. And then in 2018, there was a group of journal publishers who took action and created a roadmap for publishers defining how data citation should be handled in the context of journal publication. So with these principles, let's take a look at what benefits we actually have of data citations. So first and foremost, I would say that by citing data, we acknowledge its importance and we give credit to the data holders and publishers. And this in turn also serves as feedback to data publishers to show that their data is useful and also has an impact on science. We also increase transparency by showing that the data that underlies the conclusions of our work, and we allow for reproducibility and also facilitate the reuse of data. So I'm going to put on my GBIF glasses now and take a look at what we have been doing around data citations in GBIF. So first of all, to those of you who might not be completely familiar with GBIF, I will just briefly mention that we are an international network and data infrastructure aimed at providing anyone, anywhere, open access to data about all types of life on Earth. GBIF is funded by the governments of our country participants, and we currently serve more than 90,000 datasets from 2,100 data publishers. And in total, we have more than 2.5 billion species occurrences available. And these are downloaded at a rate of more than 160 billion records per month. This data has been used in close to 10,000 peer-reviewed journals so far. So GBIF provides access to mainly 4 different classes of datasets. We have on one hand taxonomic checklists, and these are basically lists of named organisms organized by a specific taxonomy, geography, or any other theme. Then we have species occurrences, being records of where species occur in time and space. We have sampling events that are, they provide means for more quantitative monitoring according to standardized protocols. And then finally, we also have metadata-only datasets that allow people to publish descriptions of resources that exist but have yet to be digitized. All of these are assigned unique DOIs so that they can be cited easily. Here I have just included a few examples of such datasets and how they are supposed to be cited when used in a paper. And as you can see, they look slightly different, but they all share the fact that they have a DOI in there. So occurrences are the most used type of data available through GBIF, and GBIF data users typically don't come to the GBIF website looking for information. Well, they typically come to the GBIF website looking for information about where species occur, and they don't typically care too much about how that data is organized. So we provide them with an easy means of filtering and downloading exactly the data that they're interested in. And that new unique data set that we then produce is also assigned a unique DOI when downloaded. So this is an example of what such a download might look like. I realize this might be a little hard to see, but suffice it to say that the download DOI will resolve to a landing page that has information on how to cite it, which filters were used when creating the download, how many records are included. It also has a link to be able to download the data. And importantly, it also has a list of all the source data sets. So that is the data sets from which the data in the download was compiled. So when the data downloaded is then used in a publication, we ask the authors to cite the DOI of the download. This information allows us to count the citation of the download as a citation of each of the contributing data sets. And the publishers of these data sets can then easily track how often their data is used and by which papers. So here's an example of what that might look like in real life. So here's a paper on forest herb species and how they respond to climate change. And this has used and cited a number of GBIF downloads. If we pull up one of these, you will see all the details of the download, including the list of source data sets. And in this case, this data set included data from, or the download included data from the Bush Natural History Museum. And one record was contributed to the download. But we can also see that this particular data set also has 287 other citations. And this can then be accessed for the publisher as well here. I apologize. Oh, sorry. Here we go. So there are in some cases users are able to obtain data from GBIF by other means than simply going to the GBIF website. This might be advanced users that are writing their own scripts for accessing data directly through GBIF APIs, or use existing software libraries that also access data directly through APIs. We also share monthly snapshots of eligible data on certain cloud computing environments, such as the Microsoft Planetary Computer and Amazon Web Services. And this allows for very large analyses, typically working on some filtered subset of data. But due to the synchronous nature of all these scenarios, we are not able to directly assign DOIs in these cases. So the question is, how do you then cite data? So a few years back, we introduced what we refer to as the derived dataset concept. And this is basically a metadata record similar to a download, but without the filters and without the actual data. So it's defined by the user themselves, and they need a gbif.org account to log into GBIF and then provide a summary of the GBIF datasets that contributed record to their filtered dataset, and ideally specifying how many records each dataset contributed. They can also include a URL to where their filtered dataset can be accessed. This tool, the tool to create these derived dataset records is available on the GBIF website, but it can also be accessed as a service through an API or through the RGBIF R package. So just to illustrate how this could work in real life, a user has somehow ended up with a data set of occurrences from GBIF. Doesn't matter how they got it, but the data set includes the metadata that allows the user to identify the source or the sources or data sets in GBIF. So the user is then supposed to summarize these records to get counts of how many records came from each of these parent data sets. And once they provide that, we then give the service then gives them a new DOI representing that specific derived data set. So this is an example of what a derived data set might look like to someone who accesses the DOI. And you will notice an immediate similarity to the download page, but the main difference being that the metadata is provided by the user themselves. When the user then includes a derived data set in a publication, just as was the case for citing a GBIF download, we can then resolve that citation into the relevant data sets in GBIF and award them each one new citation to update the count and the list available to the data publishers through GBIF. Okay, so let's assume that you've used GBIF data in a study and you are writing up a manuscript. What exactly do you do? Okay, so if we assume fairly standardized structure of a scientific paper, you would be including a reference to the GBIF data in the materials and methods section, ensuring that this points to an entry in the reference list with the DOI assigned to this data, just as if you were citing another paper, basically. You can, however, also choose to include it in the data availability statement if that is how your journal prefers to do data citations, or you can do both. And in all fairness, I would say that it is the citing of the data using a DOI that is the most important part, not exactly where or which style it is done. And I've just included a few examples here to show you that it does vary, but in the end, these are all fine. So there is a reason, however, why the location of data citation does to some extent matter. And that is because journal publishers are able to mark up citations, as they also do with citations of papers, so that they can become indexed and machine readable. And this in turn allows for automatic discovery and locking of citations, for example, within the GBIF ecosystem. But I would also say that this is something that falls more under the responsibilities of journal publishers rather than data users. And as a data user, if you put a citation in the materials and methods section and you reference a DOI in the reference list, you've done all that you can do, basically. The rest is up to the journal publishers. Speaking of data users, we do sometimes have very eager downloaders who reach out to us asking how to cite thousands of downloads when their journal of choice limits the number of references to, say, 50. So the first thing we tell them is to consider how data is downloaded, at least for the next time. We do allow up to 100,000 species or names in a single download. And if you only have one download, you only have one DOI to cite. In these cases, however, the damage, if you will, has already been done. The analysis has been completed. The manuscript may even have been submitted, and they need a solution right now. So we can tell them that they can either attempt to summarize the combined downloads into a single derived data set, if they're able to do so, or as a last resort, include a list of downloads as a supplement, or put it in a separate repository to which they can then provide a link in the paper. So we've been doing data citations for quite a while, and it might be interesting to just take a step back and look at how things have evolved over these last 9 years, basically. So first of all, we know that use of GBIF data continues to increase at around 20% each year. And currently, we are logging around 5 new papers relying on GBIF-mediated data every single day of the year. So as I mentioned in the beginning, GBIF started assigning DOIs to datasets and downloads in 2015. but the uptake of citing these has been fairly slow. We started using a more or less automatic workflow in 2017, in which an email would go out to corresponding authors of papers that use GBIF data, but failed to cite them using a DOI. And this has been fairly intense effort, but it seems to have paid off, as we can see that people who have been contacted seem to learn their lesson and get it right the next time. And all in all, we are seeing DOIs cited. We see DOIs used in citations of GBIF data in just over 60% of the papers that we log. So another interesting fact, especially from the perspective of data publishers, is that they're able to monitor continuously how and where their data is being used. So I've just pulled up here a graph showing the 5 largest occurrence data sets in GBIF at the moment, also showing the number of citations on the secondary y-axis. And as you can see, there's no clear correlation between size and use, if any, maybe an inverse one. However, if we take a closer look at the same data sets and instead count the number of unique taxa contained in a data set, we seem to be able to spot a trend. So in terms of impact of data, size doesn't matter, but diversity appears to matter. So just as I wrap up my talk, I wanted to mention some initiatives around improving the quality of data citations. As I've already mentioned, we're continuing to automatically notify all authors who fail to cite a DOI when they use GBIF data. We're also starting to see an increased interest from journal publishers who want to improve their workflows and guidelines, and then they actually come to us asking for guidance. We are considering whether we should be solely focused on featuring journals that encourage and enforce proper data citation, or perhaps also name and shame journals and publishers who fail to live up to their own policies and continue to basically allow bad citations to be published. Finally, I wanted to mention a new set of best practices that came out as part of a paper just a few months ago, led by Shelley Stahl from the American Geophysical Union, with co-authors from major publishing houses, including Wiley and Elsevier, as well as funders and other academic societies. This paper is another attempt to draw attention to the importance of citation and also software. The paper includes a checklist, which I've actually pasted into this slide here, mentioning how journals should provide clear instructions to authors, have proper policies in place around data citation, educate, and this is very important, I think, their editors and staff, also improve machine actionable representation of citation, and this is to allow for automated linking and discovery, as I mentioned earlier. And finally, also establish metrics to ensure that there are consistent results for data citations. I think that this is a great step, and I truly hope that journals will start following these best practices so that we may eventually get to 100% uptake of DOIs in citations. So with that, I thank the organizers for the opportunity to talk today and everybody on the webinar for your attention. Thank you.
Thank you so much, Daniel. That was really, really interesting. And can I ask Hanna to come back as well? Oh, thank you. Hi, Hanna. That was really, really interesting. Thank you both so much. We have a couple of audience questions, if you don't mind. The first of these is, the 100,000 species is the limit to download, but some is this the number of registers or individuals? That's come in the chat from Carlos Zamora.
Yeah, that's very easy to answer. This is a limit on names, so species names. There's no limit on the number of actual records that can be included in a download.
Okay, and then another question from the audience is, how do you know when an author hasn't cited properly with the DOI?
Yeah, that's an extremely good question. So one thing that I did not mention or didn't cover really is the way that we actually, the work we do to actually track all these citations. Because I could speak about that for another like 30 minutes probably. And so ideally, if citations were all done properly and they had DOIs and they were marked up and indexed and everything, we could just sit back and watch them come in automatically. Since that's not the case, we actually do a lot of manual searching, relying on email alerts using Google Scholar and similar services, and to go through basically all mentions of GBIF in papers to see if there's actual use, and then how they are citing that.
Okay, that is a lot of work.
Yes, it is.
That is an awful lot of work. Maybe we do a third webinar on just how that process works. Okay, we've got more questions coming in, and I'm going to jump back to those, but I do have a question for both you and Hanna that I also did want to ask you. So I'm taking the right to ask this question because I was very curious. What are the ethical considerations in data citation for biodiversity research? Would either of you like to give me a comment on that? So I think the idea of ethics is really, really important. And as you know, the BioDT project, we're all about the FAIR principles, we're all about this kind of thing. So ethics really needs to be considered. So what would I'd like from both of you actually to hear, what do you think the ethical considerations for data citation are in this area? Thank you.
So if I can start, I think Daniel can then give more detailed answer, but I think for biodiversity data, especially the question is that it's normally not shared through metadata catalog, but as data, and that makes it quite difficult to refer back to the researcher. So traditionally it comes from museums and bigger institutes, and referring back to the data source is easy, but giving credit to the researcher, him or herself, is more difficult. And I think the download DOI is a very good approach in this. And second thing, making sure that the researcher gets the credit is to publish data papers, so that then you as a researcher get credit for creating a data set and describing it as richly as possible, and the quality. And then there are also ethical considerations about the reproducibility of research, and nowadays with the FAIR principles, also the data part of research should be reproducible and transparent to the peer reviewers. So having the data, being able to refer back to it as it was as raw data, and also giving identifiers to the process or workflows or models or other components that are not tabular data, but they can be considered being part of data, is one question that I think is not solved yet completely, but we are getting there, and I think there's lots of good work going on at the moment in the FAIR principles, and all the research communities trying to implement FAIR.
You want me to add something? Okay. Yeah, no, I agree completely. And I would just say that generally, like anybody who, like, as part of good scientific practice, and if you acknowledge the importance and the significance of biodiversity data, using it in your research, you are morally obligated to acknowledge and to credit the people that are responsible for making the data available to you. And of course, you should also aim to increase transparency, reproducibility, and facilitate reuse. And then also, like, from a slightly different angle, at least in sort of terms of GBIF data, legally speaking, users of GBIF data are actually also bound by a data user agreement that strictly stipulates that users have to publicly acknowledge the data publishers whose biodiversity they've used, and they have to do so by citing a DOI. So even though it's very, very difficult to actually enforce this, it is actually a requirement. It's not just a good idea, it's the law.
It's not an option, you have to do it.
Yeah, yeah.
Okay, that's very, very clear. Thank you both. We have many more questions coming in, so I'm just going to jump to the next one from the audience. This is from Simon Rolfe, and Simon says, I see how this works with respect to a static published paper with one run of the workflow and a derived dataset DOI. However, in a digital twin, when models could be run daily, how do we cite the data? A new derived DOI every model run, or? Okay, I'm going to leave that to you guys to answer because that sounds very difficult.
It is very difficult, and I don't think we yet have an answer. I think because the data to biodiversity digital twin comes from 4 different distributed infrastructures, we are only able to kind of refer back to the data originator, and then if we create something new, derived data sets and models and workflows, I think at this point the approach would be to be as transparent as possible, and then refer to the processes that we have created as kind of research outputs that need to be able to cite. So yeah, I don't think we are able to be more precise than that, but this is also, it's a pilot, so we will learn and hopefully find some solutions on the way. I don't know, Daniel, if you have some GBIF insight to this problem.
Yeah, it's a really good question. I don't think I have any immediate sort of ideas for that.
That's
yeah, the
enough.
problem is that GBIF has implemented this system, and it's maybe the most advanced at the moment, where you can actually refer at the data level to the data sources, and then the rest of the infrastructures still need to implement something. So because most of them are still only working through metadata catalogs and giving identifiers to metadata records. So this is definitely a place where we need to collaborate.
For sure.
Absolutely. And, you know, we're always coming up with new things we need to be able to reference. So it's part of the practice of research. Okay, there was one question in the chat that I can answer myself, and that was whether it would be possible to see the webinar afterwards in some place. I can confirm, yes, we will be uploading the recording of the webinar to the BioDT YouTube channel, and we will also be making the slides and presentations available on the website. If you want, you can also register for the upcoming second data citation webinar, so you will have even more information if you want that. And okay, going back to questions for Hanna and Daniel. An anonymous attendee asks, how does GBIF track and ensure proper attribution for data downloaded through online tools like GeoCat, which facilitate easy access without requiring user authentication? So how do you track when they aren't requiring there?
Yeah, that's an excellent question, and the answer is that we don't. We're not really able to. So in the examples I gave of these sort of synchronous data access examples where people are just pulling data on the spot and then providing some sort of data product based on that, we don't have any means of basically linking that to the specific data used. So what we're trying to do is work with the people that create these tools to ensure that they have sort of built that into their workflows as well. If not, they might not be able to work directly and create a perfect citation, but at least make it easier for the users then to use, for instance, a derived data set to create a citation that will allow us to acknowledge data publishers properly.
And another question from the audience. if I use the protocol of data downloading suggested in the GBIF data blog using the RGBIF package, the DOI will be automatically uploaded to my profile. So
Yes.
Yes. Okay.
Yes. If you authenticate, which you will be required to do to initiate a download, the download is logged to your account.
Okay. And then I'm going to ask just one other question that you did slightly cover in your presentation, but just to really drive this point home, what are the potential consequences of not properly citing biodiversity data, both for researchers and data contributors?
So I think that for data publishers, it's crucial. Without citations, they are really not able to demonstrate, the impact of sharing data. I mean, they can only kind of guess, but so if they can't show this, it might lead to, decrease in funding and eventually, less ability and desire to share data openly and freely, and then we all lose. And I think for the researchers that use data, if they don't cite properly or use proper data citations, I think that they also are at a risk of not being able to fund and publish their research, with more both funders and journal publishers actually requiring, fair, open data cited in publications. So I don't think there's any way around this, really.
No, absolutely.
I could also add the idea that this is the same as writing a scientific paper and not citing those papers that you have drawn your ideas from. So it's just simply a scientific practice that you have to cite it if you use ideas or data from others. And I'm hoping peer reviewers will also pay attention to this increasingly, I guess.
Yes, actually, that brings me to another question. That is, how does data citation in biodiversity research differ from data citation in other scientific fields? And that's a question to both of you. So who wants to go first?
I can go because I already mentioned it, that the biodiversity data, there are other fields of sciences or disciplines where data is shared as data and not through metadata catalogs. But I think this is the kind of two ways of citing data. You either have a fixed data set that is a data paper, or then have cumulative data set, but there normally that's often also referred to via the metadata. And then biodiversity data is difficult because you can't really search or find data without the species, because you can't list all those hundreds of species that you are interested in. So it's easier to search it on the data level, and then you have to cite it as data set that you are using and extracting from the huge big data source that GBIF is aggregating globally. Maybe Daniel can explain this from GBIF viewpoint.
Yeah, I was actually I was just thinking about how data citation practices differ in other fields, and I'm not sure that it does differ significantly, but I do think that especially the GBIF community being responsible for enabling a lot of biodiversity research, I think we played a very active role in trying to cement the importance of proper data citations. And I think we've been fairly successful. And I also think that we are seen as somewhat of a role model in this aspect. But I also hope that the work that we are doing, although we're focusing on GBIF data, I hope that it will have an impact on data citation in general, and not just in biodiversity, but also in other fields.
that makes sense. And I guess there was one more question on, we also have a comment from Dag Anderson. For a live data stream, I think we need to consider a DOI for the data stream endpoint itself. Only snapshot data from the data stream could be cited as a static data set. Okay. It's very, okay. This is definitely an area that needs further consideration, and thank you for that, Dag. Okay. One more question in my list, and that is if there are any specific requirements you want to highlight or standards for citing data collected through the GBIF platform. So Daniel, is there anything else you want, other than what you've said? You've said a lot about this already. So is there anything else you'd like to add to that one?
I want to, well, no, I mean, we don't attempt to police styles or formats of citations, also because they are to some extent journal dependent. But we just insist that citations include the DOI. So if that's like, if there's one take-home message, it should be that. Always include a DOI in the citation. Yeah. Okay.
That comes to the end of the list of questions I have from the audience. And in case anyone wants to get one more question in before we finish up. And I just want to remind everyone that we do have a second data citation webinar. This is going to be Mastering Data Citation, and it's going to be insights from the BioDT research infrastructures. So that's all 4 of our research infrastructures. GBIF will be back again, but also we will have Disco, eAlter, and LifeWatch, all giving different insights from their perspectives. So today has been an absolutely brilliant presentation from Daniel. Thank you so much for providing the GBIF worldview of this, because it has been really, really interesting. Hanna, thank you so much for introducing the BioDT project. They were both absolutely brilliant presentations, and I'm very grateful. Everyone, if you didn't have a question answered today, you can use the contact form on the BioDT website to send me the questions for next time, and we might be able to cover them. I will be posting the recording of this webinar on our BioDT YouTube channel, and of course the presentations will be available on the website as well. So thank you to everyone who joined today, who asked questions, who registered, and especially thank you so much to Daniel and Hanna for their excellent presentations. So if there's no other questions coming in, with no further ado, I might close everything. Unless Daniel or Hanna, would either of you like to say anything to the end?
Thank you for having us. Yeah, it was a blast. Thanks.
Thank you. Oh, thanks to you. Daniel?
Yeah, thanks, Joanne, and thank you for letting me, giving me the opportunity to talk today.
It was really interesting. Okay, I'm going to close that here. And thank you both, and thank you everyone for attending today. Take care. Thank you. Bye.